In this video, we will discuss about the specificity and reproducibility of the multiplex system and how their multiplex system can be used to test different types of peptide, and how they have tested their system is shown in this panel A, that they have created a pool of peptides from the protein that ended at the same position, but they have a different length. So we can see that in the pool 1, the peptide that is shown on the top row is longest, and with as we go to the different rows, we can see the decrease in the peptide length, but it ended at the same position. And the examples of the proteins that they took for their studies is pro-insulin, or we can say that immature insulin, so which thereby undergoes cleavage to become the mature insulins. So they have considered this immature insulins and created a pool of peptide and for that they created many pool of peptides that we will see its results. And thereafter, this pool of peptides, since these pre-pro insulins can be used to trigger T cell 1E6 T cell receptors. So they used 1E6 T cell receptor systems in these experiments, and first they check the single fluorescence reporter 8XNFAT reporters, and they can see that the insulins that are made from, like, the peptides that are used from 12 to 24 will show a highest stimulation index. And we have understood in our previous video about the stimulation index that it is the ratio of the signals that are observed from the stimulated T cells compared to the resting T cells. So when they used it for their studies, they can see that it is for the first time they see results of around, and the peptides in the range of 12 to 24 they see a highest stimulation index, and when they do it for the second time they see the similar results. So this shows its reproducibility. Thereafter they checked it for the multiplex for the different fluorescence. So first they check for LO plus TM. In that case also they see the stimulation index for the same peptide range is 12 to 24, though it is looks like less than 20, but in the literature, in the manuscript, it is written that they have seen the highest stimulation still 12 to 24. And they checked it for this LO plus TM multiplex signalings, and then they used the other fluorescence combinations, that is AM plus CR. In that case also they see the stimulations index high from 12 to 24 peptide range. So this indicates that their reporters that they have made for activated T cells is robust and gives the high reproducibility that they have shown using this pre-pro peptides, the peptides from the pre-pro insulins, and they have used it for this single fluorescence 8XNFAT reporters, and also for the dual or multiplex 8XNFAT reporters for the different fluorescence channel, and thereby they just validate their, or they, from these results they shows the efficiencies of the system that they have developed, that is 8XNFAT reporters that can be used to generate signals for the activated T cells. So when the T cells get activated, this 8XNFAT get dephosphorylated in the cytoplasm, and this dephosphorylated NFAT then localizes into the nucleus, where they helps the transcription of the plasmid, as the plasmid contains this 8 NFAT binding sites, and thereby once the transcription of the plasmids get accelerated because of the NFAT reaches to the NFAT binding sites on the plasmid, then it will help in attracting this RNA polymerase II for the transcriptions of the plasmid, and thereby we will see a reporter proteins get translated and transcripted and thereby translated, and we will see the signals. So this is what the gist of the reporters, and they have validated its specificity and reproducibility in multiplex systems. So I hope you understood the experiments and their idea behind their reporters. If you need to understand more about their experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.